Welcome to today's lecture on molecular biology and its applications in medicine. In this comprehensive script, we will explore various topics ranging from basic principles of molecular biology to advanced techniques in genetic research. We will also delve into the genetic basis of neurological disorders and the potential for targeted treatments. So, let's begin our journey into the fascinating world of molecular biology. The study of molecular genetics in neurological disorders focuses on understanding how genetic variations contribute to these conditions. It involves investigating gene alterations and their effects on protein function, inheritance patterns, and potential therapeutic targets. This knowledge is essential for personalized medicine and improving patient outcomes in neurology. In summary, genetic inheritance patterns can be categorized into Mendelian and non-Mendelian inheritance. Mendelian inheritance follows dominant and recessive traits, while non-Mendelian inheritance includes codominance, incomplete dominance, polygenic inheritance, and sex-linked inheritance. These patterns are important for predicting genetic disorders. Molecular biology has developed advanced techniques to analyze DNA and its processes. Polymerase chain reaction, PCR, amplifies DNA, aiding disease diagnostics and genetic fingerprinting. DNA sequencing identifies mutations in genes related to neurological disorders. Techniques like FISH and microarrays visualize genetic abnormalities and gene expression patterns. Genetic mutations, such as point mutations, frameshift mutations, and chromosomal rearrangements, can impact protein structure and function. These alterations can range from harmless variations to severe genetic disorders, including neurodegenerative diseases. Studying the effects of various mutations helps us better understand the molecular basis of neurological disorders. Epigenetic modifications refer to changes in gene expression caused by modifications to DNA or associated proteins without altering the underlying DNA sequence. These modifications can be heritable and can result from environmental factors or cellular processes. In the context of neurological disorders, epigenetic modifications are of great significance. They contribute to the development and progression of disorders such as Alzheimer's disease, Parkinson's disease, and autism spectrum disorders. Understanding how these modifications alter gene expression patterns provides opportunities for developing targeted therapies. Genetic variation affects disease susceptibility and progression in neurological disorders. Single nucleotide polymorphisms, SNPs, and structural variations contribute to increased vulnerability. Understanding these variations helps develop personalized treatments. Identifying disease-causing genes is a complex process that involves large-scale genetic studies, such as genome-wide association studies, GWAS. GWAS analyze the genomes of thousands of individuals to identify genetic variations associated with specific diseases. Furthermore, advances in next-generation sequencing, NGS, technologies have revolutionized the field. Whole exome sequencing and whole genome sequencing enable the identification of rare mutations responsible for neurological disorders. This knowledge helps in designing targeted therapies and improving patient outcomes. Genomic medicine aims to integrate genetic information into healthcare to improve disease diagnosis, prevention, and treatment. In the case of neurological disorders, knowledge of the underlying genetic factors allows for tailored treatment plans. Genetic counseling plays a crucial role in guiding patients and families affected by neurological disorders. Genetic counselors provide information about the genetic basis of the disorder, inheritance patterns, recurrence risks, and available testing options. This empowers individuals to make informed decisions and better manage their health. Neurodevelopmental disorders, neurodegenerative diseases, and neuropsychiatric conditions encompass a wide range of disorders affecting the nervous system. Molecular biology provides insights into the underlying mechanisms of these disorders. For instance, studies have revealed that mutations in genes involved in neurodevelopmental processes can lead to conditions such as autism spectrum disorder and intellectual disabilities. Similarly, defects in protein clearance pathways, mitochondrial dysfunction, and abnormal protein aggregation play crucial roles in the pathogenesis of neurodegenerative diseases like Alzheimer's and Parkinson's diseases. Several neurological disorders are directly caused by mutations in single genes. Examples include Huntington's disease, Fragile X syndrome, and Duchenne muscular dystrophy. Understanding the genetic basis of these disorders enables the identification of therapeutic targets and the development of potential gene therapies. Targeted treatments aim to address the underlying genetic defects and alleviate disease symptoms. Complex neurological disorders, such as schizophrenia, bipolar disorder, and multiple sclerosis, involve the interaction of multiple genes and environmental factors. Unraveling the genetic basis of these disorders is challenging but crucial for improving diagnostics and treatment. Genome-wide studies have implicated various genes and genomic regions in the susceptibility to these disorders. Additionally, epigenetic modifications and gene-gene interactions contribute to the complexity. Research in this area continues to shed light on the underlying mechanisms and potential therapeutic targets. Advancements in gene therapy hold promise for the treatment of neurological disorders. Gene therapy involves the introduction of functional genes or genetic material into cells to correct genetic defects or modulate disease processes. In recent years, gene therapy approaches utilizing viral vectors or nucleic acid-based therapeutics have shown encouraging results in preclinical and clinical studies. Targeted treatments aim to correct underlying genetic abnormalities and restore normal cellular function, offering hope for patients with previously untreatable neurological conditions. In conclusion, molecular biology has revolutionized our understanding of genetics in neurological disorders. Identifying disease-causing genes, genetic testing, and targeted therapies provide opportunities for individualized treatment and enhanced outcomes. Let us embrace these advancements for better global patient care. Thank you for being a part of this journey.